Now we are starting chapter 15, specific immunity and immunizations. Specific immunity, also known as adaptive immunity, is the harder of the two immunity chapters. I'm including it first because we'll have exam 3, and then after exam 3, non-specific immunity is going to review a lot of the concepts that we've covered in chapter 15. So you'll get an opportunity. So if we do this first, you'll have an opportunity to review it when we do non-specific immunity before the final exam, and then we'll review it yet again when we do microbiome. So this is the hardest of the next 3 chapters, and I want to include it first because it gives us more opportunity to review several times before the final. So immunity means you get a disease, and then you don't get it again. People observed that once you get it, the concept of immunity has been noted for centuries. It's been noted for centuries that people build immunity to diseases after their first encounter. People observed, for example, that once you get a disease like smallpox, if you survive it, you almost never get that disease again. Smallpox was actually the first disease that people used vaccines for. There was this physician who first of all, there was this old wives' tale that if you got cowpox, which is a closely related but much less lethal disease, old wives' tale, you get cowpox, you won't get smallpox. But it wasn't tested, it was just this old wives' tale. And then there was this physician who went around infecting small children with cowpox to protect them from smallpox, and it did indeed work. So it was sort of an experiment. There's maybe some ethical problems with that, you might say, but the fact of the matter is that it worked, and the children who had gotten cowpox first were protected against smallpox, which is more lethal. So the use of cowpox as a vaccine against smallpox predated germ theory by 100 years. People had no idea why it worked. They had no idea why you didn't get a disease again if you had it once, or why closely related diseases protected you from each other. There are 2 main branches of our immune systems. There's the innate immune system and the adaptive immune system. The innate immune system is also sometimes called the non-specific immune system. This is the one that we will cover after exam 3. But I want to introduce it here because it actually comes before the adaptive system in terms of what a pathogen encounters. So a pathogen in your body will encounter the innate immune system first, the adaptive immune system second. So this is so the innate immune system or the non-specific immune system gives you a non-specialized response to pathogen invasion. And what I mean by non-specialized is I mean that the response is always exactly the same. It does not vary by pathogen. So anything that gets in your body that's not supposed to be there, the innate immune system gives the same response every time, no matter what it is. So that's why it's also called the non-specific response. It's not specific to individual pathogens. This immune response is less effective than the adaptive immune system, but it's fast acting. It is a very immediate response. So the innate immune system is there to stave off infection until the adaptive immune system has time to kick in. Now the adaptive immune system, also known as the specific immune system, is highly specialized. In the adaptive immune system, you have a response that's tailored to each individual pathogen. The adaptive immune system is far more effective than the innate immune system at fighting disease, but it takes many days to develop, especially the first time you encounter a new pathogen. So the first time you encounter a new pathogen, you don't have immunity yet. When we talk about immunity, we're really talking about the adaptive immune system. So if you don't have immunity to a pathogen, your body has never encountered it before, it can take a very long time for your adaptive immune system to kick in. Your innate immune system is protecting you in those first days, those first weeks until the adaptive immune system launches its full response. So there are places in which these 2 immune responses overlap. The innate immune system can trigger the adaptive immune system, and the adaptive immune system can trigger the innate immune system, especially through the complement system, but also through communication signals called cytokines, which we will cover in this lecture. So we're going to focus now on the adaptive immune system branch. It comes after the innate immune system. It's more complex than the innate immune system, but we're going to cover it first so that we have more time to review it before the final exam. So looking only at the adaptive immune system, you have two responses. You have your primary response and your secondary response. Your primary response happens the first time your adaptive immune system ever encounters a pathogen. It's a brand new pathogen. Your body's never seen it before. This primary response, during this primary response, you have peak antibody production 10 to 17 days after the infection. So the pathogen gets into your body. It takes 10 to 17 days, so about 2 weeks, to reach peak antibody production. So that's a long time, right? Your innate immune system is protecting you during those first days. Your secondary response refers to any subsequent encounter with the same pathogen. So, and your second is after so during your primary response, your adaptive immune system is generating immunity to protect you against future infections from that pathogen. So anytime that pathogen enters your body in the future, you then have a secondary response to it. It's a much faster response due to the chemical memory that your adaptive immune system generated during the primary response. So the first time you encountered the pathogen, took a long time for your adaptive immune system to gear up, but you generated a chemical memory. This chemical memory allows your adaptive immune system to give a much faster response the second time you encounter the same pathogen. Peak antibody production for the secondary response is only 2 to 7 days after infection. So a fraction, sometimes a tiny fraction of the time it took during the primary response. This lecture, so this is split up into several videos. This first video, we're going to focus on vocabulary. So I'm going to give you a brief rundown of all the terms in this slide, and then there's another couple slides with vocabulary words as well. So I'm just going to explain what each of those vocabulary words means, and then in the future videos, we will really start integrating all these vocabulary words with each other to describe what's actually going on in the adaptive immune system. So bear with me. The adaptive immune system is very complex. There's a lot of circular learning that has to take place. It's difficult to describe one aspect without having described another aspect because it's kind of like a web. All of these different facets of the adaptive immune system are webbed in a network together. And so we kind of need to, so you kind of need to know what all the pieces are before I can weave them together for you. All right, so let's start with antigens and antibodies. So an antigen is a molecule that you find on the surface of a foreign particle that's entering your body. So for example, an antigen could be a molecule on the surface of a bacteria. So it's not part of your body. It's something from the outside. It's part of an invader. It's not the whole invader, so you don't say that the bacteria is an antigen, but certain molecules on the surface of the bacteria would be antigens. An antibody is something that your immune system makes, so it's something native to you. So an antibody is built by your immune cells to fight pathogens. Excuse me, I'm going to sneeze. Nope, it left. So it's not a cell. An antibody is not a cell. It's a protein that's excreted by white blood cells. So you can think of them as weapons. These white blood cells manufacture these weapons called antibodies and then excrete them out into your body fluids. And if they bind to a pathogen, they help fight that pathogen. So think of an antibody as a weapon. It's not alive, just like a weapon for a war is not alive. The soldier's alive. The soldier's like the white blood cell. The antibody is like a weapon, like a gun or something like that. So antibodies recognize antigens. And actually the word antigen means antibody generator. So the antigen is the foreign object. And when it's in the body, it stimulates white blood cells to generate antibodies to fight the antigen. Okay. So antibodies will recognize antigens. And these different things here, the paratope, the variable region, constant region, VC region, these are all anatomical features of an antibody. So let's go into these in future slides. So let's look at the parts of let's break down antigen a little bit more. So, like I said, the antigen is a molecule on the surface of a pathogen or some foreign particle in general. And the piece of the antigen that an antibody binds to is called the epitope. So one microbe can carry several epitopes. Even one antigen can carry several epitopes. So on the surface of a pathogen, you have an antigen. The antigen can be a very, very large molecule, and it can have several binding sites for antibodies, and each binding site is called an epitope. So some places where you might find an antigen on a particular microbe would be embedded in the capsule, embedded in the cell wall, embedded in the cilia or flagella, the outer membrane. And then also protein. Usually antigens are unique proteins, so they're proteins that are embedded in those features. So it would be different from one species of bacteria to another. It's not the peptidoglycan of the cell wall, because many bacteria have that, but it might be some protein that's embedded in the peptidoglycan cell wall that is unique to one species of bacteria. So that's another thing, is antigens are unique. They're different across species of bacteria. Antigens can also be toxins that the bacteria excretes. So in the case of Clostridium tetani, which causes tetanus, when you get a vaccine for tetanus, you're generating antibodies not against the bacteria, but against the toxin that bacteria produces. Remember, the Clostridium species produce these really, really potent toxins, often neurotoxins. So those toxins are what you're generating antibodies against, and that toxin is considered an antigen, even though it's not attached to the bacteria. Other things that can be considered antigens are egg whites, pollen. The reason we consider, so these are things that in some people generate an allergic reaction. So we consider them, even though they're not actually pathogenic, we consider them antigens because they can cause certain people to generate antibodies, to generate an immune response. So here's a diagram to help understand all that. So here in yellow you have a pathogen, and then on the surface of this pathogen you have antigens in dark yellow. So this full large molecule, that's an antigen, and you can see that they surround the pathogen all over the cell surface. This little piece here, this little binding site on the antigen is called the epitope. So here's a B cell, one of your white blood cells, and it's producing antibodies. So these little Y-shaped purple things, those are antibodies. Those are the weapons produced by the B cell. So the B cell is producing antibodies to fight the pathogen, and they bind to the epitope, which is on the antigen, which is on the pathogen. And then here's another diagram showing the same thing. So here's an antibody, and here's the antigen. This piece of the antigen that the antibody binds to is called the epitope. Conversely, on the antibody, the part of the antibody that binds to the antigen is called the paratope. So the paratope on the antibody binds with the epitope on the antigen. All right, so again, the paratope is the part, so now we've moved on to antibodies. This transitions us into antibodies. Another word for antibody is immunoglobulin. I want you to remember both because immunoglobulin actually includes two different molecules, antibodies and B cell receptors, which we will get to later. So an antibody is a kind of immunoglobulin. And you need to know immunoglobulin because when we talk about different, you might have seen IgM, IgE. These are different kinds of antibodies found in the body. So they're indicated, they're abbreviated Ig, immunoglobulin for short, and then they're lettered. That could be IgA, IgG, IgE. So those are all different styles of antibodies. So remember, on an antibody, the paratope is the part of the antibody that binds to the epitope. The valence is the number of paratopes, or the number of binding sites a particular antibody has. So I was just saying you have IgEs, IgMs, IgAs. These all have different numbers of paratopes. Different antibodies have different numbers of binding sites. So bivalent is the normal, what we would say is the normal structure for an antibody. It's like the stereotypical antibody. So it's a Y-shaped. So a Y-shaped antibody is bivalent because there's two binding sites, one on each tip of the Y. So, and that's the simplest. That's the simplest version of an antibody. We sometimes call them monomers. In an individual antibody, there's a stem. The stem activates the immune system, other aspects of the immune system. And the variable region, which is the V part at the tips, that's specialized. That's where you find the epitope, and that's specialized, and it varies from antibody to antibody. So remember, I was telling you that the adaptive immune system is highly specialized, right? The reason it's specialized is these immunoglobulins, they have epitopes that are unique to each individual pathogen you have ever encountered in your life. So every time you encounter a pathogen, your body is going to start generating immunoglobulins that are special. They have these specialized binding sites that only bind to that one pathogen. So these are going to differ. The variable region where the epitopes are, that's going to vary from one antibody to the next, and it's specialized to pathogens that you've encountered. And then the stem is the constant region. The stem does not vary because its job is to communicate with immune cells. So if an immunoglobulin, like an antibody, binds to a pathogen on one end at the epitope, the constant region has to then bind to an immune cell to activate it. So let's look at a picture here, a diagram. So here's a stereotypical antibody. It's bivalent, meaning it has two epitopes, and those are in orange. And you can see in orange, it's called the variable region, because this is going to be unique to different antibodies, because each antibody is going to be specialized for a different pathogen. And then here in blue, you have the constant region at the stem. That's the piece that communicates with your immune system. So it's always the same, because it's always communicating with the same immune system. So I want to talk a little bit about how you get, about the specificity of the adaptive immune system. So we said it was called the specific immune system, and it's highly specialized. The reason is because those epitopes, those binding sites for pathogens, are unique, and they're also very diverse. So for immunoglobulins, like B cell receptors and antibodies, there are 10 billion unique shapes of binding sites. 10 billion unique epitopes that your body can make to bind to any kind of pathogen you could imagine. T cells, so B cell receptors are produced by B cells, but you also have T cells, of course. T cell receptors also have binding sites for pathogens, and there are 1 quadrillion unique kinds of T cell receptor binding sites. So between T cell receptors and B cell receptors, there is basically no pathogen that your immune cells can't bind to through these receptors. So now the genes for T cell receptors and immunoglobulins are not so abundant. There's only a few hundred of them. So how is it that you can have only a few hundred genes for your T cell receptors and your immunoglobulins, but you can have 10 billion different B cell receptors and antibodies and a quadrillion different kinds of T cell receptors? Well, the reason is these genes mutate really rapidly, and they also, there's little segments of them rearrange rapidly. So you get these random rearrangements of nucleotides coding for the variable regions of the T cell receptors and the immunoglobulins. So the rearrangements include, like I said, a high mutation rate. So the genes for T cell receptors, B cell receptors, and antibodies include a high mutation rate. But we also have recombination and mitosis. So that's called mitotic recombination. So we didn't cover mitosis in this class because we focus on bacteria. So I'll remind you a little bit of what it is. Remember, mitosis is clonal cell division. It's the way your human cells divide, right? When you're not making sex cells. So when you start out in your mother's womb as just a few cells, they divide and divide and divide and divide and divide clonally through mitosis to create, grow and grow and grow, creating a fetus and then a baby and then a child and then an adult. Also, every time you need to repair tissue from a wound or your cells die and need to be replaced, that's all done through mitosis, through clonal cell division. So usually in mitosis, you just copy the DNA and then divide it up into the two daughter cells. Genetic recombination is something that we do through meiosis, when we're generating sperm and egg. Sperm and egg, remember, are genetically unique. Every sperm is unique, every egg is unique because of genetic recombination. And what happens, if I can remind you from your prereqs in genetic recombination, is you have the father's chromosomes and the mother's chromosomes, right? We have 23 from dad, 23 from mom. They pair up with each other in one part of meiosis, and then certain pieces of mom's chromosome will pop off and switch places with the homologous segment of dad's chromosome. So these little pieces of chromosomes from your mom and your dad can pop off and switch places with each other and shuffle. And that's how you get genetic shuffling. So then when the DNA is divided up into egg or sperm, it's all shuffled uniquely every time. So this usually genetic recombination only happens in meiosis, in the production of gametes. But in these particular genes, for immunoglobulins and T-cell receptors, we get genetic recombination all the time. So whenever a stem cell that's producing B or T cells divides, or whenever a B cell itself divides to make offspring and a T cell divides to make offspring, those particular genes are engaging in genetic recombination, meaning the nucleotide sequences are shuffling between the maternal and the paternal chromosomes, creating new unique sequences every time they divide. So you have the mutation rate and you have mitotic genetic recombination, which creates diversity, which means that these genes, even though there's only a few hundred, they keep changing all the time. So even though there's only a few hundred of them, they keep changing, keep shuffling, and so you can get this 10 billion different kinds of immunoglobulins and a quadrillion different kinds of T-cell receptors. So an analogy to conceptualize this, you have more than 1 million English words from only 26 letters. Remember, you can get all these different words because even though you only have 26 letters, you can arrange them differently to make different words. So it's the same idea with these genes. So we will cover this in the coming videos, but I'm going to introduce the idea now. If it sounds confusing, don't worry, you will get diagrams and everything of this. But I know that at this point, a lot of students are wondering, how is it, if you have all of these different kinds of immunoglobulins, how do you make the right immunoglobulin or the right T-cell receptor for a new pathogen that comes in? I always get that question really early, so I want to address it now and tell you that we will go into more depth about this later. So the way that happens is you're actually not making antibodies and T-cell receptors for a pathogen that you've encountered. What you're actually doing is making random shapes of T-cell receptors and B-cell receptors and antibodies. So every time you make a new B-cell receptor, it's going to be a random shape because of this genetic recombination and mutation. It's just, they're randomly changing all the time. So every time you make a new B cell, every time you make a new T cell, they're randomly creating new, unique T-cell receptors, B-cell receptors, and antibodies. Then, if one of those randomly generated B-cell receptors binds to a pathogen, it's going to make more of itself. Then it'll make true clones of itself. So if one of those randomly generated B-cell receptors or T-cell receptors proves useful in binding to a pathogen, they're going to make a massive army of clones of that exact B-cell receptor or that exact T-cell receptor. And those clones will live in your body for years and years and years, maybe even a lifetime. So what happens is you're randomly generating receptor shapes. The ones that prove useful get copied and they stay in your body. And that's what gives you immunity. So the useful ones proliferate and become dominant. So like I said, we'll come back to that a few times before these lectures are over. So there's this important aspect of immunity, or this important process of generating B and T cells called clonal deletion and thymic deletion. Clonal and thymic deletion, so I just told you that your B cell receptors and your T cell receptors are randomly generated. What if some of them just happen to bind to molecules that are normal in your body that you need, like proteins on the surface of your own cells? If that happens, your B cells and T cells would start attacking your own body, and that would be called an autoimmune disorder. So to prevent that from happening, your B cells go through clonal deletion and your T cells go through thymic deletion. In clonal deletion and in thymic deletion, the B cells and the T cells are tested before they're released into your body. And they're tested to make sure that they don't bind with proteins on your normal cells. If they do, they're destroyed and they're never released into your body in the first place. So that prevents autoimmune attack. In the case of thymic deletion, 98% of T cells are deleted and never released into your body. So you have all of these T cells being generated in your bone marrow, maturing in your thymus, and 98% of them get deleted because the receptors on their surface could bind to your own cells and cause an autoimmune attack. So they're deleted, meaning they're induced into apoptosis. So here are all of these different immunoglobulin shapes. So you have IgG, IgE, IgD, IgM, IgA. I am not going to make you memorize. So the way these different antibodies vary is in part by shape. So you can see some of them look different from others. They have different valences or different numbers of binding sites. But another way they vary is in function and where you find them in the body. So some you're going to find in your gut more often. Some you're going to find in bodily excretions like saliva or breast milk. I am not going to make you memorize that. It's easy to look up. I just want you to notice that these are different and the different shapes. So you have the bivalent, which is normal. So there's the bivalent means you have 2 binding sites or 2 epitopes, two paratopes. And then IgA here it has a valence of 4. It has 4 binding sites or 4 paratopes. IgM has a valence of 10. Again, I don't need you to memorize which is which. I'm just showing you how these vary so that you can understand. Because when you read literature about the immune system, you're going to see IgG all the time or IgE or whatever. And I just want you to know what that means, generally speaking. It's just a different kind of antibody. All right, so moving down in the vocabulary list, B cell receptors and T cell receptors. So we already sort of covered this, but so T cell and B cell receptors are very much like antibodies in that they are specially designed to fit one type of epitope from a pathogen. But they are not released into what makes a B cell receptor and a T cell receptor different from an antibody is that they are not released into body fluid, while antibodies are. So a B cell makes an antibody and excretes it, and then the antibody flows around in your body fluids, your blood, your lymph. B cell receptors stay stuck on the surface of B cells. T cell receptors stay stuck on the surface of T cells. B cell receptors are really like antibodies. Actually, they are antibodies that are not released. So a B cell receptor is just an antibody that has not been released from the cell surface yet. Okay? So that's why B cell receptors and antibodies are both referred to collectively as immunoglobulins. They're the same molecule. So you have this one kind of molecule called immunoglobulin. When an immunoglobulin is embedded in the surface of the B cell, it's called a B cell receptor. When the immunoglobulin is released into the body fluids, it's called an antibody. All right, so here's a diagram of what they look like. So here's the B cell receptor, and you can see it's just an antibody, but it's stuck in the surface of the B cell. T cell receptors, here's a T cell receptor. It has this binding site to recognize pathogens, and it's embedded in the surface of the T cell. And then there are these other receptors that are adjacent, and we'll get to these down the line. But they are for cell-to-cell communication within the immune system. So the T cell receptor here, it's binding to a pathogen. These cluster of differentiation, we'll come back to them later, but just know that there are these molecules here to help the T cell communicate with other immune cells. Because you have so many different kinds of immune cells, I'll just go back to this slide. You have so many different kinds of immune cells, and they're constantly communicating with each other. PAMPs, or pathogen-associated molecular patterns, TLRs, or toll-like receptors, PRRs, or pattern recognition receptors. These vocabulary are really important to the innate immunity chapter. I'm going to introduce these concepts here, but they are not going to be on this next exam. It's convenient to mention them while we're covering antibodies, B cell receptors, T cell receptors, and antigens. So a PAMP, or a pathogen-associated molecular pattern, is like an antigen. It's a molecule that you find on the surface of a pathogen. What makes a PAMP different from an antigen is that antigens are unique and PAMPs are not. So an antigen is a molecule that is unique to one species of bacteria or one species of virus or one species of fungus. A PAMP is a molecule found on the surface of pathogens that's shared by many, many different kinds of pathogen. So for example, a PAMP might be peptidoglycan. All bacteria have peptidoglycan. So that's an example of a PAMP. Peptidoglycan is not unique. All bacteria have it. A toll-like receptor and a pattern recognition receptor, those are both similar to B cell receptors and T cell receptors. They're found on the surfaces of immune cells, but of immune cells that are part of the innate immune system. And their job is to recognize PAMPs. So B cell receptors and T cell receptors recognize antigens. Toll-like receptors, pattern recognition receptors, recognize PAMPs, or pathogen-associated molecular patterns. So like I said, you don't need to take notes on this now. I'll bring this up after exam 3 when we cover innate immunity, but I just want to mention that they're here because it makes it easier later. MHC, there are so many acronyms in the immunity chapters, so I'll just try to repeat them as often as possible. So MHC stands for Major Histocompatibility Complex, and you have MHC1s, MHC2s. MHC1s, Major Histocompatibility Complex 1, and Major Histocompatibility Complex 2. Class 1 MHCs are on every cell of the animal host. You can think of them as the name tag that says self or host. And they are required so that the immune cells don't go around destroying its own team. So if your immune cells begin to attack your own cells, right, that's like we said, that's an autoimmune disorder. So one way that that could happen is if a pathogen, is if there are some pathogens that disrupt your MHC class 1s. So some viruses, when they get inside your cells, they knock out the MHC class 1. Then your immune cells don't know that your host cell is your host cell, and so they can attack it. So I would like you to think of MHC class 1 as the self tag that indicates I'm supposed to be here. MHC class 2s you find only on the surface of immune cells. Okay, so they are like little display cases for antigens on immune cells. So there are certain cells in your body that phagocytize pathogens. So these are certain white blood cells whose job it is to go around your body looking for foreign material like pathogens. Once they encounter foreign material, they phagocytize it, they ingest it and digest it. Once they do that, they can display the heads of their enemies, as one of my students called it. They can display the heads of their enemies or an antigen, a molecule from that pathogen that they just digested, on their MHC class 2. They're displaying it there so other immune cells can find it. So other immune cells, if they find an antigen on MHC class 2, it will activate them and they'll say, oh, there's a pathogen. This immune cell just digested a pathogen. That means that there are invaders. We need to all become activated now and also go search out the rest of those pathogens. So MHC class 2s are display cases for the heads of enemies. So here's a diagram displaying this. So this would be an immune cell because it has not only MHC class 1s that all cells have, but also MHC class 2s. So MHC class 1s have more than one job. So and I want to mention to you, there's 2 colors of each, and that's because you have one from your mother and one from your father. So you have the red and green. The red is the maternal MHC class 1, so that's the self tag inherited from your mother. In green, you have the paternal MHC class 1. That's the self name tag inherited from your father. And then you have the MHC class 2s. And the MHC class 2s are only immune cells have these. So that indicates that this is a kind of immune cell. So in blue, you have the maternal MHC class 2. In gray, you have the paternal MHC class 2. And this is, so when this immune cell in green ingests a pathogen and digests it, it's going to then take the digested material and display it here on the MHC class 2s to show to other immune cells to activate them. One of the reasons organ transplants are really difficult is because I just showed you here, different people have different MHC class 1s or different self tags. So when you get an organ transplant, the donor does not necessarily have the same MHC class 1s as you do. And so your immune cells are programmed to recognize the ones that you were born with as normal. Foreign ones it won't recognize. So it's not going to be able to, when you receive an organ transplant, if the donor didn't have the same MHC class 1s, your body is going to say, I can't find the self tag because it doesn't recognize the other person's self tags, and it'll attack the organ as a foreign object. And that's why we suppress the immune system when we give organ transplants, so we can suppress the autoimmune attack. All right, phagocytosis. Phagocytosis is a very specific form of endocytosis. It's how a single cell eats. So some immune cells, but not all, can gobble up pathogens via phagocytosis, and we call those kinds of cells phagocytes. That's not how all immune cells work. Different immune cells have different jobs. The ones that are able to ingest and digest a pathogen, to phagocytize a pathogen, we call them phagocytes. So phagocytes phagocytize. Antigen-presenting cells, or APCs, they are special kinds of phagocytes. So APCs are phagocytes that have MHC class 2s for presenting the heads of their enemies to the other immune cells. So some phagocytes ingest and digest, and that's the end of it. Some immune cells, like APCs, ingest and digest, then take some of the digested material and display it on their MHC class 2s to other immune cells to activate them. So regular phagocytes don't communicate with other immune cells. Antigen-presenting cells are phagocytes that do communicate with other immune cells about what they just attacked. B cells are a kind of antigen-presenting cell that we'll cover in this chapter. But there are other antigen-presenting cells that belong to the innate immune system that we'll get to in the next chapter. So here's a phagocyte. This phagocyte is gobbling up this bacteria. And this works through receptor binding. So here you have the cell surface, this blue line. You have all these receptors, like perhaps B cell receptors here. There's a pathogen, and the pathogen has antigens on its surface. When the antigens from the pathogen successfully bind with the receptors, for example, the B cell receptors on the surface of the phagocyte will then be stimulated to ingest that pathogen. So first it reaches out pseudopodia, and the pseudopodia reach out like arms and engulf the pathogen to bring it in via endocytosis. And here's another diagram of that. So you have bacteria that bind to receptors on the white blood cell, on the phagocyte. That stimulates these pseudopodia, or false feet, to reach around and engulf the bacteria into what we call a phagosome. The phagosome is just the food vacuole where you find the pathogen now. The phagosome is going to fuse with lysosomes. Remember lysosomes from cell anatomy, the cell anatomy chapter? Those are the digestive organelles. They're little tiny vesicles that are filled with digestive enzymes. So the phagosome is going to fuse with the digestive organelles, the lysosomes. The lysosomes then spill their digestive enzymes onto the pathogen. The pathogen is digested. Oh, by the way, when the lysosome fuses with the phagosome, we call it a phagolysosome, which is easy to remember. So then in the phagolysosome, digestion occurs. And then for a regular phagocyte, these digested particles will just be exocytosed, so they're just spit out. But for an antigen-presenting cell, some of that debris isn't spit out. Some of it's presented on the MHC class 2s to activate another immune cell into action. We're almost done with the vocabulary here. So cytokines. Cytokines are a general term for small proteins that are involved in molecular signaling. They have several important functions in the immune system. So you can think of them as the communication molecules of the immune system. almost all white blood cells can produce cytokines, and they allow the different white blood cells to talk to each other, to activate each other. So they're just the communication molecules of the immune system. So here's some things that cytokines can do. Cytokines are extremely diverse. So this is not an exhaustive list of functions, but these are just a few styles of cytokines that I would like you to know, because we'll keep coming back to them in the innate immune system and in the microbiome chapter. So cytokines can induce stem cells to become T or B cells. So if one of your cells has encountered a pathogen, one of the things it might do is release cytokines, which send signals to your bone marrow, where the stem cells for your blood are, and signal the stem cells in your bone marrow to become more white blood cells, like more T or B cells. Cytokines can attract T cells and other immune cells to a site of infection. So if one pathogen encounters, if one white blood cell encounters a pathogen, it might release cytokines as a chemical trail to attract other white blood cells to that site where the battle is going on. Cytokines can initiate inflammation. We usually think that pathogens cause inflammation, and so inflammation is a bad thing, but actually inflammation is your body's response to infection, and it helps you fight the pathogen. It helps you get rid of the pathogen. Additionally, it's not on here, but I should say inflammation and fever. Those can go hand in hand. They're usually caused by similar cytokines. So cytokines can cause inflammation. They can also cause fever. Cytokines can also interfere with viral replication within cells. So sometimes cytokines are released to stop a virus from replicating inside your cells when you get infected with a virus. All right, and the final two words here are humoral immunity and cellular immunity. So at the beginning of this chapter, we talked about how there are two types of immune systems, the innate and the adaptive immune system. Zooming in closer into the adaptive immune system, there are additionally two branches of the adaptive immune system, the humoral and the cellular. The humoral immune system targets pathogens in the extracellular space, the outside of cell space. And so that means in the body fluids. So your humors are your body fluids. So the humoral immune system is attacking pathogens that are floating around in your body fluid. But remember, when we were discussing mechanisms of pathogenicity, we talked about how some pathogens try to evade your immune system by hiding out inside your human cells. So some pathogens will try to evade or hide from the humoral immune system response by entering into your normal host cells, and where the humoral immune system can't find them. And that's where the cellular immune system comes in. So the cellular immune system targets the pathogens that have entered your human cells that have so they're hiding from your humoral immune system. So the cellular immune system can find them. Both humoral and cellular immunity involve a memory of the disease. We discussed how that immunity is generated a bit before, so we're going to review that in a bit more detail here. The humoral immune system's main actors are B cells. Remember, B cells produce immunoglobulins, like antibodies, but also B cell receptors. So they have B cell receptors on their surface for binding antigens on pathogens. These B cells and their immunoglobulins target pathogens invading extracellular space, outside the cells, in the body fluids, or sometimes on the surface of host tissue. The cellular immune system's main actors are T cells. T cells have T cell receptors on their surface to bind to antigens and pathogens. Like immunoglobulins, there are a baffling diversity of shapes, of receptor binding shapes, allowing T cells to target very specific antigens on very specific species of pathogen. Just like B cells, the main difference just like B cells, but the main difference is that the cellular immune system has the unique ability to target intracellular pathogens, pathogens that are hiding inside your cells, like viruses and some bacteria. T cells also play an important role in activating many parts of the immune system through cytokine signals. So cytokines are really important to the cellular immune system, to T cells. And remember, cytokines are those communication molecules of the immune system. So T cells can talk with and activate many different kinds of immune cells through cytokine signaling. Right, and we will stop here and pick up on diving into detail on these two branches of the adaptive immune system in the next video.